O-Level Biology 5090, Paper 2, Variant 2, October, November 2024, question number 6 and 7. Let's move towards the question number 6. And question number 6 is, farmers in many countries, including the United States of America, USA, keep cows to produce milk. This milk is then sold for human consumption. So table 6.1 shows how the yearly average milk production per cow in the USA changed over a period of 60 years. Table 6.1 shows that from 1950, it was 2250 cow per kilogram per year. So 1970 it increased 4500, 1990 6750, and 2010 it has the milk production has been increased per cow, it is 9400. So between the years 1970 and 2010, a farmer in the USA always had 250 cows producing milk on this farm. Use the data in table 6.1 to calculate the increase in milk production by 250 cows between the year 1970 and 2010. Express your answer in standard form, space for working. So we have to write as it was. We have to find out the data, the correct answer between 1970 to 2010. So in 1970, the milk production was 4500 kg per year, and in 2010 it increased 9400 kilogram per year. So it means we have to find out first the increase in milk production by 250 cows. The data which has they have given us in the form of table, it is for one cow, and we have to find out for about 250 cows. So first of all, we have to start from 1970. 1970, the milk production was 4500, and in 2010 it increased 99,400, and then we will increase in milk production. Is 9400 minus 4500. So it is equal to 4900 per kg for one cow. But we have to find out for about 250 cows. We will multiply 250 with 4900, and we will get the value after calculating with calculator. It is equal to 1,225,000. But this is the answer, but we have to write in standard form. So standard form will be, we will write 1, 2, 3, 4, 5, 6. So we will write 1.225 into 10 raised to power 6. So we will write in this way the answer. So this is the answer in standard form per kg per year for 250 cows. Now we are moving towards the next question, and next question is the part 2 of the A. One reason for the pattern shown by the data in table 6.1 is the use by farmers of artificial selection to breed cows that produce more milk. This is an example of selective breeding. So use this example to describe the main stages in the process of selective breeding. So first of all, we must know what is selective breeding. Selective breeding, also known as artificial selection, which is a process in which the humans intentionally breed plants or animals for particular desirable traits. In the context of increased milk production in cows, you can see we have to uh, link it with the previous question in which they were producing the cows for more milk. So for first of all, for this process of selective breeding or artificial selection, there should be the identification of the desirable traits. The persons who are going to do this, or to they are the farmers, first of all, they should identify the what they want. First of all, as they need the high milk yield, so they will find out that animal or the cow uh, which has this quality, that is it is producing the uh, high yield of the milk. So first point in this regards, we can say that is the identification of desirable traits, or identification of the desired of desired traits in a cow who is producing high yield of milk, or giving more production of the milk. And secondly, uh, then that cow can be used as a parent to reproduce the um, organisms. So the second, we can see select the cow as a parent to reproduce the organisms. That cow which is selected should be the true breeders. True breeders are those organisms which give the same results after many generations. So they are producing only the desired characteristics, or the desired requirement is there in those types of cows. Then after that, you can see it will involve in the case of cow, this involves mating the high milk producing cow with carefully chosen bulls. So this allows for the combination of desired genes from both parents to be passed onto their offsprings. So this cow, mating of this cow with the bulls of good quality. After their mating, you can see the last step is the selection of the offsprings, that the which offspring is according or very close to the desired or the required. So the offspring produced from this breeding are then evaluated for the desired traits, and then farmer would observe the milk production of the new generation of cows. So those offsprings that show an even higher milk yield than their parents are then selected to become the parents of the next generations. Then at selection of the offsprings for the next generation. That is, who are yielding the, who are giving more production of milk than their parents. So these experiments were then last repetition of these processes to get more production. In short, I would like to say the increase in milk production in cows is, as illustrated by the data, is a direct result of farmers using selective breeding to enhance their specific desirable trait over several decades. So they identify higher producing cows, bred them, selected the highest product producing offsprings, and repeated this process, leading to the substantial increase in milk yield we observe. So in this way, we can see that this process can be used for producing the high yield, the cows which are giving the high production of milk. Now the next question is, research has shown that 95% of cows used to produce milk in the USA are fed crop plants that have been produced by genetic modification. These crop plants have been produced to provide the cows that eat them with additional vitamins. Outline the stages in the genetic modification process used to produce these crop plants. So I think the same process is involved as, for example, that this time we have to give the outline of the stages in genetic modification. Genetic modification, is also a process of producing the offsprings, high quality offsprings, which are genetically modified, and the characteristics are according to our desire. So in the first step is that we have to identify the of organisms which have the desired genes. So first step is identification. Identification of the organisms or plants with the desired genes. Desired genes means with additional vitamins. With vitamins. So after their identification, there will be the isolation of those genes. So it needs to be cut out or isolate from the organism's DNA. So this is typically done using specialized enzyme, which is called as restriction enzyme from plant's DNA by restriction enzyme. And then insertion of these genes into the vector. into the vector. And you know, vector is a carrier that can deliver the foreign gene into the plants, and most commonly used vector in plant genetic engineering is a plasmid. And plasmid, you know, part of the bacteria. Circular DNA, bacteria, it contains circular DNA, which is called as plasmid. It can easily be obtained from the bacteriums. So bacteria is used for this purpose. So it is inserted into the vector by an enzyme ligase. Then after its insertion, the vector containing the desired gene is introduced into plant cells. Then vector is introduced into the plant cells by specific means. And then you can see at the end, we can see the transgenic, those plants are called as genetically modified plants. And will express the desired traits. So in this way, these crop plants can provide cows with additional vitamins. So these steps are involved in this process of genetic modification. Next part of the question. Genetic modification has been used to change crop plants in other ways. One example of this is the production of crop plants that contain a chemical to make them resistant to insect pests. Resistant to insect pest. Uh, Outline two potential risks of growing these crops. That is, if you can say the crop plants are resistant to the pest, to the insect pests, then outline two potential risks of growing these crops. So you can see, always we know there are two views of everything. That is, on one side, this is good for, it has advantages. On the other hand, it has uh, disadvantages as the same one. So in this way, you can see, uh, if we introduce chemicals into specific plants for and to make them insect resistant to the insect pests, so there is a risk that non-target insects, beneficial including beneficial insects like pollinators or predators of other pests, could also be negatively affected. So exposure to the chemical, even it is less potent on them. So I will development of resistance. So the one effect can be the development of resistance in non-target insects which are beneficial for plants. So on the other hand, they cannot be the beneficial for it will uh, not help the plant. And there can be second point can be the gene flow to wild relatives. That is, and that is, there is possibility there is possibility that the genes which are introduced into the crop plants could be transferred to other wild crops which will show the negative effects towards the environment and ecosystem. Because they will negatively affect, because always we want to reduce these wild crops which are not beneficial for the healthy plants. So in this way, if the genes are transferred to them, the genes with the insect resistance, so if they are transferred to them, then it will give negatively affect the environment. So these are the 2 points. Now, the next question is question number 7, which is very simple question. Is we have to compare between different processes. First of all, the difference, diffusion and active transport. So active diffusion and active transport, these are the 2 processes by which the different molecules move from one side to the other, or one area to the other area. So first of all, we have to discuss about diffusion. Diffusion is a process in which the molecules net movement of the molecules from the region of high concentration to a region of lower concentration. and this movement is down the concentration gradient. On the other hand, the active transport movement of molecules movement of molecules from a region of lower concentration towards the high concentration by using energy. Or this movement is against the concentration gradient. So there is difference in the direction of the movement. Diffusion from high to low and active transport low to high. And when the movement of molecules is from low to high or against the concentration gradient, they need energy in that case. But diffusion is a very slow process in which there is no need of energy. And in this way, you can see membrane is required in case of active transport, but in diffusion, membrane is not required. So in this way, these are the two types of definitions of diffusion and active transport. Now the next is transpiration and translocation. Next is transpiration is the process of water movement through a plant and its evaporation from aerial parts. You can say transpiration is the evaporation of water molecules from aerial parts of plant. The movement is unidirectional. That is from soil to the upper parts plant. And in this you can see the water is moved from the soil to the through root hair cells. Is it absorbed through root hair cell by the process of osmosis, and then after absorption and water enters into the xylem vessels, and from xylem vessels uh, the water is moved in the upper direction through the xylem vessel in the leaves, and then through leaves stomata it is evaporated into the air. Whereas you can see the translocation is the process of transporting organic substance, or you can see translocation is the process of transport of organic molecules. For example, glucose, which is manufactured in green leaves by photosynthesis. By photosynthesis. So from leaves towards all other parts of plants, movement is in both directions. Movement is in both direction, but already told you that in case of movement of water, transpiration, the movement is unidirectional. That is, as you can see in the plants, the water is moved in the upward direction as transpiration. Whereas in this way, but in case of translocation, the glucose can move in any direction because it has to reach those parts of the plant where it is needed, as for example, flower bud, root tips, shoot tips, where the growth is taking place. So it has to store in different parts of the plant. So in this way, the movement in all directions, but in transpiration, the movement is only in upward direction. Now, next question is phototropism and geotropism. We are moving towards that next question, which is phototropism and geotropism. Actually, these are the type of movements of the plants which takes place as a result of different light and gravity of the earth. First of all, the phototropism. Phototropism is the growth response of plant towards or away from light. Light stimulus. In this case, light acts as a stimulus. So, you know, the shoots move towards light, show positive phototropism. Whereas roots move away from light, show negative phototropism. On the other hand, the geotropism or gravitropism, so is the growth response of plant to the force of gravity. Gravity is the growth response of a plant to the roots shows positive gravitropism because their direction is towards the soil or towards the earth or gravity, whereas shoots show negative gravitropism, which move away from the gravity. So in this way, these are the different definitions, or we had to compare them. So you need to know about them, you need to learn about them, and for your exams, best of luck for your exams, and please like, share, and subscribe my channel. Thanks for watching.